Here we're going to be covering the small intestine histology. The small intestine is the digestive system compartment that is right after the stomach. Most digestion and absorption of nutrients will occur in this long tube that's called the small intestine, which will extend from the stomach. It will coil through the abdominal cavity, as we see over here, and eventually it will open into the large intestine. The small intestine is going to be divided into three regions: the duodenum, the jejunum, and the ileum. The way I remember is that I think about DJ ileum, because I usually get confused the jejunum and the ileum. So just think about DJ ileum and you won't forget the order. Now the duodenum or duodenum is this initial portion of the small intestine. It is going to be separated from the antrum of the stomach, or the pylorus. Antrum and pylorus are the same thing, by the pyloric sphincter. So that pyloric sphincter, which is located at the end of the antrum of the stomach, will separate the stomach from the small intestine, more specifically from the duodenum. The jejunum is the next portion of the small intestine, and it's going to be connecting the duodenum to the ileum. And the ileum is going to be the final and the longest portion of the small intestine. Now, the wall of the small intestine is going to be composed of the same 4 layers as the rest of the GI tract, with several modifications. Therefore, it will contain the following layers from the most superficial, meaning it is the one that's going to be closer to the lumen, to the deepest. So we're going to have the mucosa, the submucosa, the muscularis externa, and the serosa. With regards to the modifications, we can clearly see the presence of these mucosal extensions that are called plicae, and these plicae, they will have on their surface further membrane extensions that are called villi. And these villi might present even further extensions that are called microvilli, so smaller villi. All of these extensions are to increase surface area. And why would you want to increase the surface area in the small intestine? Well, in the small intestine is where most of the absorption occurs, where most of the nutrients will cross the digestive epithelial lining and enter the blood vessels to be carried to the other parts of the body. Therefore, by increasing the surface area, it makes the absorption even more effective. And in fact, it is able to increase the surface area by as much as 20-fold. Notice that within the villi, we also have the presence of the lacteals, which we have already talked about, and we will talk furthermore in the next few slides. We will still have the presence of the nervous plexus, that it's present throughout the digestive tract. So the myenteric plexus will be present right over here between the circular muscle layer and the longitudinal muscle layer. And the submucosal plexus, like the name says, will be present within the submucosal layer. Even though the duodenum is the shortest subdivision of the small intestine, measuring only between 8 to 10 inches long, it has the most features of interest. It receives digestive enzymes from the pancreas via the main pancreatic duct, which is this duct right over here, and bile from the liver and the gallbladder via the bile duct, which is this duct right over here. Now these ducts, they will enter the wall of the duodenum, where they will form a bulb that's called the hepatopancreatic ampulla, also known as ampulla of Vater. Now this ampulla opens into the duodenum via a mound that's called the major duodenal papilla, which can also be called the papilla of Vater or the sphincter of Oddi. Now the entry of bile and the pancreatic juices that's coming from the pancreas into the duodenum is going to be controlled by the sphincters of the smooth muscle that surrounds the hepatopancreatic ampulla and the ends of the pancreatic and bile ducts. Therefore, the bile that's produced by the hepatocytes in the liver will come down through a right and left hepatic duct. They will merge to form what we call the common bile duct. From here, the bile will be stored in the gallbladder, passing through the cystic duct. When needed for fat digestion, and we will talk more about this later on, the bile that's stored in the gallbladder will come down through the bile duct and empty into the duodenum, passing first through the hepatopancreatic ampulla and then through the major duodenal papilla and ending in the duodenum, where the fat is going to be present because it's coming from the stomach and emptying into the duodenum. Also, as we can see from this image over here on the right, it shows the comparison between the three different tracts of the small intestine. Compared to the jejunum in the ileum, the duodenum contains numerous duodenal glands, which can also be called the Brunner's glands. And these glands, they will produce large quantities of mucus, and the mucus that's produced by the intestinal and the submucosal glands, they will protect the epithelium from the acidic chyme that's arriving from the stomach. And the mucus will also contain buffers that will help to elevate the pH of the chyme that's emptying from the stomach. Because remember, the stomach has a very low pH. Now, the submucosal glands are most abundant in the proximal portion of the duodenum, and their numbers will decrease as they approach the jejunum. And over this distance, the pH of the intestinal contents, they change from about 1 to 2 to about 7 to 8. And why is this? Why is it that these mucus glands are more abundant in the proximal portion of the duodenum? Well, remember that buffers and enzymes from the pancreas and bile from the liver enter the duodenum roughly halfway along its length. Therefore, the buffers from the pancreas will help to neutralize the pH right in the middle of the duodenum. Once the pancreatic juice enters through the major duodenum papilla, or also called the sphincter of Oddi. So there's no need for you to have large quantities of these glands once you pass this sphincter. Most nutrient absorption occurs in the jejunum. As we get closer to the ileum, the plicae and the villi, they become smaller, and they continue to become smaller in size and number as we move closer to the ileum. And this means that the reduction in the absorptive activity diminishes as well. With regards to the length, the jejunum is way longer than the duodenum. Where the duodenum was measured in inches, now the jejunum is measured in feet. So it's roughly about 8 feet long. Now, the ileum is going to be the longest of all of the structures of the small intestine. Is actually about 10 feet long. And as we can see, it has less of the plicae. It will have less villi because once we get to the ileum, there's less absorption that occurs, as most of the absorption will occur in the jejunum. But we do have these aggregated lymphoid nodules that are called Peyer's patches that will be located, especially when we get closer to the area where the small intestine will meet the large intestine. So right over here is what we call the ileocecal valve. It can also be called the ileocecal sphincter, and this is where the small intestine, more specifically the ileum, will join the large intestine, more specifically the cecum. And this is why it's called the ileocecal valve, because it's a portion of the ileum with the cecum coming together. Now, why do you think we have these Peyer's patches located in the ileum, or more specifically closer to the ileocecal valve? Well, these are going to be important because specifically in the large intestine we have large amounts of bacteria. These are normal bacteria that we need for our digestive system to work properly, but they do not belong in the small intestine. And once the ileocecal valve opens, we don't want the bacteria from the large intestine to come into the small intestine. However, this sometimes happens, and this is why these pyre patches are located in the ileum right next to the ileocecal valve to basically destroy any bacteria that backflows from the cecum into the ileum, and therefore preventing an infection from occurring. On the lab models, I have placed this image over here that will show you how the organs are organized within the abdominal cavity. So here would be your liver. Right underneath the liver is the stomach. And if we remove the stomach, we're going to find the pancreas right over here. Continuous with the stomach, so this would be your stomach, we will find the duodenum right over here. And running behind the large intestine, the top half would be your jejunum, and the bottom half would be your ileum. So again, this would be your top half is your jejunum, and the bottom half is your ileum. Same thing over here. We can see a little bit of the duodenum right over here, but the top half would be the jejunum, the bottom half would be the ileum. If we see a posterior view, which is this image right over here, we can see the whole duodenum right over here, or duodenum, and underneath we can see a little bit of the ileum. The other structure that we see on this image that we didn't really talk much about is this yellow structure over here, which is called the greater omentum. And this is one of the mesenteries that actually hangs from the stomach, and it's made up of fat tissue that will actually support and cover all the organs and the intestines that are found in the lower abdominal area. Now, it also serves as fat deposition, which will vary in the amount depending on the individual, as you can imagine. There are certain studies also that show that there are collections of macrophages within the greater omentum, so they might serve as an immune contributor. And it also prevents the parietal and the visceral peritoneum of the abdominal cavity from adhering to each other. And especially during the winter, depending on the amount of fat that you might have, it might help you keep warm.